Well, thank you, everybody, and thank you in particular for another year of your leadership with the Holiday Challenge. We're going to have a brief research update for all of you. I think you'll find it of great interest. Andrew Heaton, our Chief Scientific Officer, is joining us on this call as well as the calls over the next couple of days as well. I think you all have started your Holiday Challenge pages as I look at who's with us tonight, but if you haven't, it's curejm.org/HopeHero. And remember that gifts for the campaign, including your personal gifts, will be matched by the Coffee Family Match. So that's another real terrific incentive, if you will, for all of our donors that we've been able to accomplish again this year. So thank you so much for your support and for your leadership once again in this campaign. Andrew's going to share with us a little bit in a few moments about the sort of what I look at anyway as the sort of basic to translational, basic research to translational research to clinical trial research. That is kind of the typical path, if you will, that nonprofit research organizations like CureJM will go through over their history as they expand and as they learn about the disease and what we can do to really arrest the disease through a combination of basic research, just fundamentally understanding the disease, translational research, which is doing the kind of work you need to do to prepare for a clinical trial. That will include things like animal models, and Andrew may share with you that we now are working with two different sets of animal models as we look forward to clinical trials in new drugs. That's an important step that we're making. And then finally, clinical trials, and we have 3 clinical trials that we're involved with right now. One has pretty much wrapped up, actually. That's a trial out of George Washington University for a drug called abatacept, known also as Orencia. While that trial has finished, the data analysis, it has not and is ongoing right now, and we're hopeful that we'll see some very positive results. I think some of the early results kind of through the grapevine look pretty encouraging in terms of the effectiveness of that drug for some children with juvenile dermatomyositis. I also wanted to share with you that we've increased our grants budget this year by 1/3, and in addition to that, we have added funding for 3 new centers of excellence in areas of the country that we think areas of the country that currently are underserved, or where CureJM would like to increase its presence. I think you're all familiar with our 4 existing centers of excellence in Seattle, Chicago, George Washington University in Washington, D.C., and Duke University in North Carolina. I don't know yet where these 3 new centers will be located, as we're just now beginning to. We put out the RFP about 30 days ago, and those are due, I think, Andrew, by December 31st, if I recall correctly.
End of November.
Yeah, and so shortly thereafter, our medical advisory board and selection team will be making recommendations on those new centers, and so early in 2022, we'll be moving, I hope, from 4 to 7 centers of excellence, if in fact we end up having 3 qualified applicants, which I think is highly likely. The purpose of these centers on the clinical side, obviously some are research centers, most are research centers, but on the clinical side, the purpose of these centers is to increase the quality of care at the institutions where centers are housed by, among other things, having a more comprehensive JM program than may have existed previously, more interdisciplinary, if you will, including dermatologists, mental health specialists, physical therapists, that kind of thing, as well as training more staff within their pediatric rheumatology department on the secrets, if you will, and the challenges of treating JM effectively. So that's pretty big and expansive news for us. I'm very proud that we're able to do that, and we have been able to do that as a result of our success in previous years. So thank you for all of that. Without your past support and your longstanding work on behalf of Cure JM, we wouldn't be where we are today. It's also my pleasure, as we look at what is really the kickoff for our most important campaign of the year. It's my pleasure to introduce you to Dr. Andrew Heaton. I think some of you have met him. Many of you have heard him speak in the past. Andrew has served, I think, a little over 3 years now, almost 4 years, as our Chief Scientific Officer. He has a very impressive background in drug development. You'll notice when you hear him that he's originally from Australia, but now is happily a US citizen. And we're delighted to have him managing and overseeing our research program, including overseeing and developing and creating Cure JM's leadership role in a couple of clinical trials that I think he's going to share a little information with you about the progress in those trials. So Andrew, over to you.
Thanks, Jim. Absolute pleasure to speak to you all tonight. And Sharon, please stop me if I start going too long or I start rambling. What I wanted to give you a flavour of is some of what we've got pre-existing, and then, as Jim mentioned, some of the really exciting things we've got going forwards. So with our COE network, our Centres for Excellence, they all pride themselves on providing, the highest level of clinical care. Typically, our COEs have not just a paediatric rheumatologist, but they will also have a dermatologist that consults when patients come in. They'll have physical therapists, they'll have mental health coordinators, and they will try and organise it so that it's a one-stop shop as much as possible. And they're each pioneering their own ways for doing that. And, Duke have done some really interesting things over the last 12 to 18 months with practicing telehealth and telemedicine. Kaveh Ardalan, who some of you may know, Dr. Ardalan was previously at Lurie and has moved down to Duke. And Duke was already a very strong centre of excellence, but with Kaveh joining that team, it certainly boosted it. And I think one of the things that's a particular note with that centre of excellence is, as Jim mentioned, not only do they participate in excellence in clinical care, but their research component is very important. And with Duke, the comparative advantage with Duke is they've got a sister institute called the Duke Clinical Research Institute. So that really helps them forge a pathway forwards for clinical trials. And I think probably the most exciting thing that Duke are doing and have been doing for the last 6 months, and certainly we hope they'll be doing it for the next 18 months plus, is they've really got behind vamorolone. So many of you have probably heard of vamorolone. This is the steroid replacement that's been generated by Reveragen. Reveragen are now at the stage where they're applying for what's called an NDA, a new drug application. So it will be marketed for certain indications. And thankfully, we've got an excellent relationship with Reveragen and with Duke. So the aim is that we've got those two groups married up with DCRI, and we are hoping to put forward what's called a pre-IND meeting to the FDA. IND stands for Investigative New Drug. While the drug has been trialled before, it hasn't been trialled in JM patients. So we need to put an application into the FDA to allow us to trial it in JM patients. And the group at Duke and ReveraGen have done an outstanding job getting a clinical synopsis together for a potential vamorolone trial. And I'm hoping that that is going to be submitted literally any day, and it will get posted onto our website when it does get submitted. So that's one of the exciting things, that we have had it on a slow boil for the last little while, but it's now about to reach a critical milestone. Once we get it to the FDA, we get feedback from the FDA as to our clinical trial design. They may make some suggestions. The hope is that then Duke would then take the program and they would initiate the clinical trial in conjunction with ReveraGen and some other groups. And we're looking at innovative ways at which we could fund that via grants and a range of other things. That's not to say that our other centres aren't doing excellent work as well. I think, as Jim mentioned, GW pride themselves on those difficult to diagnose patients. And as Jim mentioned, their abatacept trial has finished. They enrolled 10 patients. That's now completed. They're just doing data analysis. Associated with GW is Dr. Hannah Kim, who works very closely with a gentleman called Dr. Robert Colbert, who are both at the NIH. They combined ran a study on baricitinib, so one of the JAK-STAT inhibitors, in 4 JM patients. They were so excited by the results, and Eli Lilly, that owned the drug, was so excited by the results that they've allowed them to expand the trial to 20 patients. So they're about to start the next phase of that clinical trial program, which is potential new medication with a JM label claim for it. So that's going forwards courtesy of GW and the NIH. So as well as Jim said, these existing centres of excellence, we're going to be looking at trying to qualify some other clinical centres that really aspire to bring forwards, the ultimate in, diagnosis and treatment, all the new treatment modalities. And we're looking forward to getting the applications in over the next little while. Besides the COEs, we're also looking at those groups and those clinicians that are perhaps just behind applying for COE status. We're expanding to what we call our clinical care network. So this is recognition of those clinicians that are, beyond the normal paediatric rheumatologist and are really have some focus and some excitement and some energy behind JM, both in diagnosing and treating JM patients. Besides our clinic network, as Jim mentioned, we're very heavily involved in primary research, translational research, and then clinical trial programs. One program that's just come to an end that's been really fruitful is the NCATS program. So that's the National Centre for Translational Studies. We had a fellow work at NCATS for 4 years. He developed a screen, what we call an in vitro screen. So he set up a model of muscle cells that mimicked JM as much as possible, as much as you can do in an in vitro setting. And he screened 4,600 drugs that are registered through the FDA. The idea being that he was screening them in an agnostic fashion. So he wasn't looking at drugs that had a previous indication for autoimmune diseases. He was looking at anything and everything. And interestingly, he tossed up a lot of the drugs that we know about already, so some of the JAK-STATs, but he also brought up some new potential drugs that may have some future traction as far as treating JM. So we're really looking forward to taking those to the next level, which brings us on to, how do you take, once you've looked at a screen in vitro, how do we then take those to the next level? One of the programs we're currently funding is with Dr. Melissa Morales up at Binghamton, and she has got a mouse model that, again, it's a genetically engineered mouse model that mimics JM, and we can test any drugs in that model. And this includes the compounds that are coming from NCATS. So what we've got now is we've got a nice train forming whereby we've identified of the 4,600 drugs that Travis tested, which ones are already active in his model. We're then picking 3 or 4 of those to take into the mouse model, with the idea being that we generate sufficient information from that data set to hopefully, convince the pharma companies that own the drugs that they should really look at starting a clinical trial in one of these newer drugs. So I think a prime example there is deucravacitinib, which is owned by BMS, which is their new JAK-STAT TYK2 drug that is just going through the end of its clinical trial program at the moment. And we're in conversation with those folks already about trying to get them excited about getting that drug into clinical trials for a JM indication. We have a bunch of other grants that are currently ongoing. Just the highlights are Dr. Jessica Tournier at Michigan. So Dr. Tournier had a previous incarnation almost in lupus, and we managed to convince Jessica that she really should become an expert in JM, which thankfully she has done over the last 2 years, and she's really hit the ground running. Jessica has a really strong interest in the skin component of JM, and she's one of the most active researchers looking at what's actually happening in the skin component. How does the disease progress? What's the trajectory? How can we treat it? Dr. Jessica Neely at San Francisco, she's got a very interesting program, again, looking at repurposing drugs and looking at the genetic signals associated with patients that are on different drugs based on the sample cohort that they have in hand. So we've got a great background of research that's currently ongoing, and I think, as Jim mentioned, it was fantastic that we were able to expand our research grant program this year, and the research committee and the board have just approved 3 new grants. So the first one is Dr. Sarah Sabar. So Sarah used to work at the NIH and had an association with GW and Dr. Lisa Ryder. So she has an excellent CV, and she's a clinician researcher of the highest order. So Sarah is now at Wisconsin. So she's set up a clinic, and her clinic is very much modeled on what GW used to do. So, we've got somebody there that is, really holding their hand up as, being a leader of the future from a clinic perspective. But interestingly, from a research perspective, Sarah has looked at JM with a different lens. And the different lens that she's looked at it is she's developing a mouse model where she's looking at the T cell populations. So we know that T cells play a critical role in all autoimmune diseases, but particularly in JM. So what Sarah is looking at is developing a model looking at T cells, Treg cells, looking at the variation in population, and again, screening a variety of drugs through that program. So we now have 2 what we call orthogonal mouse models. So we've got 2 different models. So we basically, the more models you have, the more you'll be able to filter the drugs and hopefully de-risk them and convince somebody to do a clinical trial. So Sarah's going to be working on that program for 2 years. Dr. Sarah Tansley, who's at Bath University in the UK, the group at Bath are the preeminent group as far as MSAs are concerned. So they are the leaders in the not just running routine analyses, but also developing new analytical techniques to actually measure MSAs. And I think probably, as most of you know, a lot of patients, when they do go in for an MSA analysis, quite often it will come back as, we didn't detect any MSAs. And I think a lot of the medical community are actually aware that there probably is an MSA there. It's just that we don't know what it is, and we haven't got a method for it yet. So Sarah and her group at Bath, over the next 2 years, are going back through the JM cohort of patient samples from the UK, looking at those patients where they came back as MSA negative, with the aim of trying to identify potentially new MSAs. The long-term goal behind Sarah's project is obviously, is there a correlation between MSA status and disease trajectory? Can it be used as a potential biomarker for response? So the overall strategy is to, tailor medicine to an individual patient. So if an MSA is an indicator that there may be, a significant disease trajectory, that may mean, utilising a variety of drugs early. If the disease trajectory is considered to be to be more mild, then the disease strategy would imply we use milder drugs and a milder treatment protocol. So it's really exciting what Sarah's doing at Bath. And one of the things that Sarah has done, and I think it's going to be on our website and maybe Shannon can probably post it, is Sarah and Dr. Liza McCann, who are two leading lights in the JM research community in the UK. They just recorded a podcast on MSAs, how MSAs are used by clinicians, how they're used in diagnosing patients and treating patients. So we'll provide the link to that podcast. It's very technical, but it's worthwhile listening. So I think it's about 25 or 30 minutes long. So if you're going for a run on your treadmill, or if you're going for a walk outside and you like listening to your iPhone, then I'd recommend you listening to this. You might have to listen to it a few times, but it's really a great asset for understanding MSAs. And last, but certainly by no means least, Dr. Jane McMahon is based up at Sick Kids Toronto, and she works for a group led by Dr. Brian Feldman. Now, Dr. Feldman is on our MAB, and Dr. Feldman has a global reputation in planning clinical trials and running programs where we have very low patient numbers, but he can still establish correlations from those low patient numbers. So he's a bit of a mathematical whiz kid. And what Jane is doing in collaboration with Brian is they've already had a look at some of their patient samples from the cohort of patients they've been treating up in Toronto. And what they're looking at is what we call the interferon signature. Many of you have probably heard that JM is what is referred to as an interferonopathy. So it is an autoimmune disease that's driven by this natural cytokine that goes out of control called interferon. And there are various markers around interferon, other cytokines that can help predict or potentially predict the course of disease and thereby how well treatments are working. And because there are so many different cytokines and because there are so many phenotypes of JM, nobody's been able to deconvolute the data sets into something that's meaningful for individual patients. So because we've got Brian and Jane that are effectively mathematical gurus at that, they have got a beautifully triaged data set from the patients that they have been seeing over the last few years at Toronto. They've already started work on it and have got some initial exciting results to the extent that the board thought it was very worthwhile. We actually fund this program because while it is basic research looking at interferon and other cytokines, the potential outcome from this over the next even few years is if they actually validate an interferon score for a specific disease phenotype, that's definitely going to affect how clinicians would potentially look at treating patients and utilising this interferon score. And I think, just to finish off, last year, some of you will be aware, we ran an international medical conference for new treatments for JM, which was run in collaboration with Duke. Our intention is to run one again next year, early next year in February, again in collaboration with Duke. So we get what's called continuing education credits. which means you get a lot of doctors that turn up to these conferences virtually. And when we're actually planning this conference, we will certainly be highlighting some of the research that, that we funded and some of the exciting opportunities that are coming up, just to ensure that we don't do this in isolation, and we ensure that the word is spread throughout the JM clinician community to actually establish what we've got as real groundwork for, diagnosing and treating, and I think importantly, treating appropriately our sick kids. So I think I probably have my 10 minutes, Shannon.
That was wonderful, Andrew. Thank you for that. I'm sure there's questions on the line, so maybe we could pause here and see if anyone wants to jump in and ask Andrew or Jim a question about all those great updates.
I'll sneak in for a question. So thank you very much for that. As a parent in this JM world, what do we think is the timeline for something like vamorolone if they're going through the trial? And then what's the best case scenario for when families would have access to vamorolone potentially?
So there's going to be access at a number of time points. So to go through the timeline, the pre-IND document will be going to the FDA in the next few weeks. The FDA meet, give us feedback. They will give us feedback typically within 60 days, which means we'll get feedback by the end of the year. We have already set aside a budget whereby we're going to then, the foundation will write what's called the clinical trial protocol, which ends up being a 100-page document. And we'll then be looking at funding this trial. So that trial will start initially with adults and then go into 15 to 30 JM kids. So we're hoping that that trial will start before the middle of next year. Depending on recruitment, that trial is anticipated to take between 12 months and 2 years. So the idea is that we'd enroll at least 12 adults, including a reasonable number of females. So it has to be both male and female adults, so that we can then go into male and female pediatric indication, pediatric cohort. Once we've gone into that cohort, as I said, depending on how fast we recruit the trial, and I think given our network, this trial is going to recruit very quickly. So I'm thinking it's going to be closer to 12 months than 2 years. That will end up being an initial clinical trial which will establish safety and some efficacy data points. It won't be enough to give us a label claim to the FDA, but by that stage, the drug will be on the market for Duchenne's muscular dystrophy. If we have got some clinical trial data that is published in a pediatric cohort with JM, and there's no side effects and there's an efficacy signal, and that data is published, then at the end of that 1 year to 2 year clinical trial, there is the potential for clinicians to prescribe it off-label. Obviously, there's then the typical insurance issues. And the idea is that at the end of that clinical trial, we would then look at running what we call a licensure trial, which is again another 2 year program, probably between 30 and 50 JM kids, and looking at more biomarkers of response, looking at more efficacy signals. And at the end of that trial process, it then has a label claim. So 4 or 5 years out, the drug is potentially on the market for our JM kids, but in 2 to 3 years, it's potentially available off label. And I think everybody is aware that we are going to be advertising when the trial opens and looking for people that want to enroll in the trial.
Thank you. That was excellent.
Pleasure.
Any other questions? I know we all have some as grandparents and parents. And if you think of them after the call, you can always message me and we'll get them to Andrew, or message Andrew directly, andrew.heaton@curejm.org.
All right. Well, thank you all again for being with us through another holiday challenge. As I mentioned, this is the most important campaign for us in the coming year as we look towards 2022. It's always the most important campaign of our year because success in the holiday challenge tells us what we're going to be able to accomplish in 2022. So it's, in a sense, it's like early funding for our next round of grants, and I think most importantly for the vamorolone trial that Andrew was talking about. You know, that trial, the first phase of that trial, the one where we're looking for safety and some signals of efficacy, will cost somewhere in the neighborhood of $1.5 million, maybe $2 million. That is, by clinical trial standards, exceptionally inexpensive. It's a lot of money for CureJM. You know, we're as our former chairperson, Rhonda McKeever, was often fond of saying, you know, we are the little engine that could. And in fact, you know, we are that. When you think of an organization of our size and relative youth, you know, a lot of these disease organizations have been at this for 30, 40, 50 years. We've been at it for 18 years. So when you combine those two things, our relative youth, our small size, you know, what we have been able to accomplish because of all of you is extraordinary by any standard. And as we work into these clinical trials, and as I'm confident those trials will yield not only a lot of important information, but in all likelihood, you know, the early signs are extremely positive for the two drugs that Andrew was talking about, baricitinib and vamorolone. So if in fact we see those drugs becoming available to our JM kids in the short term, 2 to 3 years is extremely short term in drug development. It will be an extraordinary achievement, much of it due to the success of our holiday challenges. So thank you for being with us year in and year out. And I look forward to talking with you again as we work through this year's campaign. And if you have any additional questions, you know, either now or shoot them to Andrew or me in an email, or Shannon as we work our way into the campaign.